Dear students, welcome back to my YouTube channel. And we were discussing about the topic applied organic chemistry. And today I would like to discuss with you some basic elementary ideas related to pharmaceuticals. And medicinal chemistry and pharmaceutical chemistry are disciplines at the intersection of chemistry, especially synthetic organic chemistry and pharmacology and various other biological specialties, and they are involved with the design, chemical synthesis, and development of bioactive molecules, that is drugs. Compound used as medicines are most often either organic compound or some biologics, small organic molecule. For example, atorvastatin and fluticasone, clopidogrel. And biological means this is often medicinal preparation prepared by medicinal proteins, either natural or recombinant antibodies or hormones. For example, infliximab and erythropoietin and insulin glargine. And next one is naming of drug. Drug nomenclature is a systematic naming and especially pharmaceutical drug. In the majority of circumstances, drugs have three types of name: chemical name, most important of which IUPAC name, and generic name, non-proprietary name. The chemical names are scientific name based on molecular structure of drug. There are various system of chemical nomenclature, and there's various chemical names of any substance. The most important one is iopicine. Chemical names are typically very long and too complex to be commonly used. For example, 1-isopropylamino, 1-naphthalenoxypropane 2-ol is a chemical name of propranolol. And during the development, company will apply for regulatory approval of the drug by various chemical agencies in that country. Nowadays, the drug that make it all the way through development, testing, and regulatory acceptance, pharmaceutical company gives the drug a trade name, which is standard term in the pharmaceutical industry for a brand name or trade name. For example, Lipitor is the Pfizer's trade name for atorvastatin, a cholesterol-lowering medication. Also, Atocor is an atorvastatin brand name in India. So for example, generic name amoxicillin, its chemical name is hydroxybenzylpenicillin, and its brand name is alpha mox or amoxil. So full chemical name is too long and complicated to use. For example, 4-amino acetaminophenol, that is paracetamol, and acetate salicylic acid, aspirin. And generic or non-proprietary name is accepted internationally as a common name. It is shorter, paracetamol and chloroquine. It is more acceptable for prescription. And brand name given by pharmaceutical manufacturer for its own particular brand or formulation. And it is followed by, for example, Panadol, Septrin, Amylar, Fansidar. These are the brand names of drug. For another example, the pain reliever, Tylenol. The brand name is Tylenol and generic name is acetaminophen. So this is the difference. Branded drugs and generic, they are produced by, branded names are, patent protection. They have company has for 20 years. After 20 years, the same company will give a generic name for that company. Generic drugs are made available at the market by the pharmaceutical company, various pharmaceutical company, after the patent protection period expires. And next one, what is meant by Prodrug concept? Okay, and Prodrug means actually the concept was first introduced by Adrian Albert to describe the compound that undergo biotransformation prior to biological effect. So prodrug, it is defined as it is biologically inactive derivative of parent drug molecule. Okay, this one that it usually requires a chemical or enzymatical transformation. This is the prodrug. It consists of a pro moiety which link to a drug and this whole part is called prodrug, and this is biologically inactive, and after entering into the body it become active by enzymatical transformation. Enzyme break this bond and it release the active drug and it possess improved delivery over parent molecule. These are the promoieties that usually use, and it link by a linker to the drug. After entering to the body, it deliver. So, for example, this is the drug design strategy. So this is a drug. When drug itself goes to some physiological membrane, it cannot enter. So what can we do is you can attach the drug with a promoiety, and which is make it easy to enter into the cell and our physiological membrane, and after reaching the body through entering through the cell, you can cleave this prodrug by enzymatic, as either enzymatic action or chemical release, and after reaching the target it can release the drug and receptor, and finally it can act. Okay, so prodrugs are compounds which are inactive themselves but which are converted in the body to the active drug. And it is useful in tackling problems like drug toxicity, bad taste, short duration of action, acid sensitivity, and poor membrane permeability. And prodrugs are activated by metabolic enzyme, for example, ester prodrug hydrolyzed by enzyme esterase. Next one, what is mean by pharmacy? Pharmacy, it is a science and technique of preparing and dispensing drug. It is a health profession that link the health science with chemical science and aims to ensure the safe and effective use of pharmaceutical drugs. Pharmacy is the health science and profession that melds chemistry, biology, anatomy, physiology, and other biomedical science together. And it is a science of drug making or deal with the procurement, testing, storage, and conversion into suitable forms. And next one, pharmacophore. Pharmacophore means it is a description of molecular features that are necessary for molecular recognition of a ligand by macromolecule. So this is the biological macromolecule, and this biological macromolecule like enzyme in our body, it recognizes some foreign particle or some substrate, for example in this case drug. So to recognize some drugs, there should be some features for that. So that study, the abstract description of features that are necessary for molecular recognition, this has to recognize this drug. And that area of study is called pharmacophore. So it is an ensemble of steric, some cases steric features are there, and some cases electronic features that are necessary to ensure optimal interaction between macromolecule and substrate, that is drug. That interaction, supramolecular interaction are there, and it is like an lock and key concept. It binds specifically, this part binds specifically to this part. So rigid lock and key concept, this is an induced fit model of enzyme substrate host-to-gastric binding. Such studies are called pharmacophore. So typical pharmacophore features include sometimes hydrophobic interaction and aromatic ring, hydrogen bond acceptors or donors, or cations and anions. So such area features have to be understood. So that area is called pharmacophore. So this pharmacophoric point may be located on the ligand itself, or may be projected points presumed to be located in the receptor. So one of the crucial characteristics of a biological molecule is the interaction, electrostatic attraction. Now suppose this is a biological acceptor and this is a drug molecule, and this biological receptor may contain various acceptor molecule and toner molecule. So we have to design a drug molecule having a toner and acceptor moiety, then which you can easily make it fit into this part like this and recognize, and it forms a supramolecular complex. For example, binding of adrenaline to receptor of G protein, which increases the blood pressure. So the binding between drugs and biological receptor by hydrogen bonding can be studied. So such an area is called pharmacophore. And what is meant by pharmacology? It is a branch of biology concerned with the study of drug action. That is pharmacology, right? And pharmacology is a branch of biology concerned with the study of drug action and study of interaction that occurs between living organism and chemicals that affect normal or abnormal biological action. There are two types. One is pharmacodynamics, another one is pharmacokinetics. Pharmacodynamics is the study of biochemical and physiological effect of drug, especially pharmaceutical drug. Pharmacodynamics is study of how a drug affects an organism, whereas pharmacokinetics is the study of how the organism affects the drug. So that is the difference. Pharmacology, pharmacons means drug, logos study, and pharmacokinetics means what the body does to the drug. It refers to the movement of drug in and alteration of drug by the body. It includes the absorption, distribution, binding, metabolism, and excretion. Pharmacodynamics means what the drug does to the body. It includes physiological and biological effect of drug. And pharmacokinetics means once the drug is injected, it goes to the stomach and the intestinal mucosa. It absorbs the, and finally it distributes all over the body through the blood plasma, and it undergoes finally metabolism in liver, and after that it excreted out through urine and bile. So these are the process that occurs, and such processes are collectively called pharmacokinetics. What is pharmacodynamics? Drug action at a receptor site and the physiological or chemical behavioral effect produced by this action. So it is the study of drug mechanism of action at the molecular level, and it provides basis for rational therapeutic uses and design of new superior therapeutic agents. Last one, pharmacognosy. It is study of medicinal drug derived from plants or other natural sources. So it is the study of physical, chemical, biochemical, and biological properties of drug substances or potential drug or drug substance of natural origin, as well as search for new drug from natural source. And it is also defined as study of crude drugs, and it is scientific and systematic study of structural, physical, chemical, and sensory character of crude drugs of vegetable, animal, and mineral origin, along with their history, method of cultivation, and collection and preparation for the market. That area is called pharmacognosy. And finally, that's all. And thank you for watching.